Hi! So in today's lesson we're going to be looking at proteins within the topic of biological molecules. So we're going to be looking at the structure of amino acids, how we form peptide bonds, the different stages of structure of complex proteins, and finally we'll be looking at how you test for proteins. But first of all, let's just think about what proteins are. So is there anything you can tell me that you think of when you think of proteins, or like any information that jumps to your mind? So they are numerous, they're very large for molecules compared to carbohydrates and lipids, and they're composed of amino acids, of which there are 20 naturally occurring. So the way that works is that the amino acids are a monomer, and the polypeptide chains that form are the polymer. And something else which is quite important about proteins is that they are all, as molecules, of a unique shape. So when you have the bonding and interaction of polypeptide chains, these form unique shapes, which is very important for some of their functions, such as being enzymes. Okay, so let's move on and think about the structure of amino acids. So I'm just going to enter the pen function and we can start drawing this out. So an amino acid, which is obviously the monomer for the polymer protein, is based around a central carbon atom. Now, bonded to this carbon atom will be hydrogen, an R side group, and that R side group doesn't stand for anything in particular. It's just a placeholder, as the side groups of amino acids are what differ between the 20 naturally occurring ones and are what give them their unique functions. So we'll cover that a little bit more later. And then really important for the structuring of polypeptide chains is the carboxyl side group, which looks like this. And then also the amino group, which looks like this. And then that is a standard amino acid. And for instance, if we had hydrogen as the side group, which would be the very most basic, that would make this amino acid glycine. Okay, so let's consider a little bit about the peptide bonding, which is what unites amino acids into polypeptides. So the peptide bonding occurs through what is called a condensation reaction. Now, that's significant because it means we're losing water. That's what happens in a condensation reaction. So the way that the amino acids bond to each other is that the hydroxyl group of the carboxyl side chain and the hydrogen of the amino group bond with each other and form water, which is then removed from the molecule. So if I just go into a different colour for a second, we can then highlight that the carbon in the carboxyl group, which wants to be forming 4 covalent bonds, will now only have 3. And the nitrogen in the amino group, which wants to form 3 bonds, will now only have 2. So the consequence of that is that the nitrogen of one amino acid and the carbon of another amino acid can bond, and that's how we get our peptide bond forming. And then eventually, when we have lots of those bonds forming in a chain, that's how we get our polypeptides. So let's move on now from the peptide bonding, and let's consider the primary structure of proteins. So what do you think we mean by the primary structure of proteins? Yeah, so the primary structure of proteins is just the chains of polypeptides. Let me just go back to black. It's the chains of polypeptides which form from these condensation reactions. So it's the chains of polypeptides. And it's really important to consider that with these 20 different amino acids, there's essentially a limitless number of combinations, which means that there's a really, as I said earlier, a numerous number of proteins that you can get being formed. And another important thing when considering the primary structure is that the order of the amino acids in this bonding for the polypeptide is determined by DNA. And that's the first of a really important number of synoptic links that we can think about within this topic as we think about the broader significance of proteins. So the first one being just the fact that it is the DNA which determines the structure of the proteins, as it is the DNA which determines the order of the amino acids. Okay, let's move on to considering the secondary structure of proteins. So the secondary structure is how we have shaping of the polypeptide chain. And what's significant about this is that the amino group has a positive charge. Not a full positive, it has a partial positive charge. And similarly, the carboxyl group has a partial negative charge. And what that means is that we can get hydrogen bonding because the partial positive charge in the amino group comes from its hydrogen, and the partial negative charge in the carboxyl group comes from its oxygen. So that means that we can get those interacting within a chain and attracting each other, and that can cause the chain to gain a new shape. So there's two main shapes that we can think about when we're thinking of the secondary structure that a polypeptide chain can take. Do you think you can name either of those? And I'll give a hint that one of those should come to mind when thinking of DNA. So that would be that we have the alpha helix or the beta pleated sheets. Now, within the content we're looking at within biological molecules, the alpha helix is one that comes up much more often as more significant. But it's good to also keep beta pleated sheets in mind, as they're very important for the structure of complex proteins and will certainly come up more if you pursue biology to higher education. So let's move on and think about the tertiary structure of proteins. Now, the tertiary structure is all about how once you go from the basic shape of an alpha helix or a beta pleated sheet, and we'll take it as an alpha helix for now, you can have more twisting and shaping of the polypeptide chain, and that can come in the form of more hydrogen bonding, ionic bonding, which will be taking place between the carboxyl groups and amino acids that aren't currently involved in a peptide bond, and then disulfide bridges. And these are significant as these will only form when you have an amino acid which contains sulfur. So for instance, you could have methionine, which has sulfur in its R side chain. So just to cover that again, in methionine, the R group here is sulfur containing. And the tertiary structure is really significant as this can be determinant in the role the protein plays. So you can have a tertiary structure where it's more round and compact, or you can have a tertiary structure where it's narrow and straight, or narrow and straight and long. So this is significant because this will play a part in the role that the protein has. So if you have a round and compact protein, we'll call that a globular protein. And if you have a narrow and long protein, that will be a fibrous protein. So this is significant because they'll then play different important roles within the body. So a globular protein will be a functional protein, and examples of those are things like enzymes, whereas a fibrous protein will be a structural protein. And an example of that would be collagen, which is important as collagen is structural in that it helps keep our skin taut, whilst obviously things like enzymes help speed up reactions. Now let's finally consider the quaternary structure of proteins. So do you have any thoughts on what the quaternary structure could be, having looked at the primary, secondary and tertiary structure? So the quaternary structure is how we have proteins and multiple polypeptide chains interacting with each other. So it's the interactions of multiple polypeptide chains to form large complex proteins. So this is really important for making these proteins complex enough molecules that they can fulfill their role that they need to in the human body. Another important thing within the quaternary structure of proteins is that you can have the introduction of prosthetic groups. And the best example of this within the human body, which is also another good synoptic link to considering transport in the body, is the heme functional group. Now, that's important as I'm sure you can probably see the link to another word you can think of, which is hemoglobin. Now, that's very important within red blood cells, where it helps bind oxygen for the transport of oxygen throughout the body. And that's possible because we have these proteins which can accommodate this prosthetic group, which is the heme, which is essentially iron in the form of Fe2+, and that's acting as a ligand for oxygen molecules. And we're dipping a little bit into the chemistry there, but that would be very useful as well for your study of chemistry. And whilst it's not directly needed for biology, it's the kind of thing that's good to know, both as a synoptic link and for higher education. Finally, let's consider how you test for proteins. So the test that we have for proteins is called the Biowrap Test. And what you have to do for this test is add a sample to a test tube. This is what you think might be the protein. You then add an equal volume of sodium hydroxide, and then after mixing you add a few drops of dilute copper to sulfate. Now, can you think where we've maybe met copper 2 sulfate before? This is another good little link to another topic, but we've met it by a different name. And that's that in the test for reducing sugars, we met Benedict's reagent. And Benedict's reagent is copper 2 sulfate. It's not necessarily good to link them too closely, as you want to use the separate names when being examined, but it's an interesting link to know how these chemicals have multiple different uses. And then finally, the diagnostic result for if we do have proteins present in this sample will be a blue to purple color change. And as always, when you're writing out a test in exam conditions, make sure to include as much detail as possible within the time. And that's really important because that means you're maximizing the marks you can get.